In this video, we will go over a step-by-step guide to the Fluid Dynamics lab. By the end of this activity, you will hopefully have a better understanding of the role that fluid dynamics play in ocean circulation and the significance this has in supporting marine biological processes in the ocean. As you will learn through this experiment, physical forces are the driving mechanisms of many different phenomena in the ocean. Take this image of a phytoplankton bloom in the Bering Sea off the coast of Alaska. Pay a close attention to the swirling eddies circled in red. In what direction are they swirling? What, if anything, is causing this motion? It's difficult to visualize this type of motion with fluids in a lab setting, so we will utilize food dye in order to see how the motion of water changes with the influence of certain physical factors such as rotation, temperature, and salinity. In this first part, we will observe how dye moves within a non-rotating tank. Pour a few drops of dye into the container far enough apart so that they don't cluster together. Watch from both an aerial and side view to see how the dye moves through the water column. How did the two colors mix? Is there structure to this motion or is it random? Is this how water moves in the ocean as well? Now we will see how the geophysical processes that inform ocean fluid dynamics are due to Earth's rotation. After the water has been sufficiently spun up, we will add a few drops of dish soap in order to help break the surface tension of the water before adding a few drops of dye to the tank. Be sure not to add too much dye as it will oversaturate the water, or too little as to not see the full effect. Once you have added the dye, observe the fluid motion and position from both above and side as the container continues to rotate. What geophysical processes in the actual ocean could be behind these movement patterns? At this point in the experiment, you will also want to add a perturbation to the water. How does this affect the mixing of the dye? As a note, depending on the size of the containers and the speed at which it's rotating, it may be difficult to see how these processes play out. Now let's take a look at the last two activities side by side. What do you notice is the difference between the motion of the dye when the tank is rotating versus stationary? How would this affect the water and nutrient circulation in the ocean? As you might have already learned, ocean circulation is not only impacted by geophysical processes, but also temperature, density, and salinity gradients. We will now examine how temperature has an effect on fluid dynamics in the ocean. In this exercise, we have placed a cup in the center of our container before it has spun up, which will be used to create a temperature gradient. You can take the center of the tank containing the ice to be the poles and the outer rim to be the equator. Start by adding ice into the cup, being careful not to spill and disturb the water. Then place 4 small drops of blue dye at or near the ice cup. As before, be sure not to add too much dye. Now place 4 drops of yellow dye closer to the outer rim of the container. The blue dye will represent the cold water in this example, and the yellow will represent warm water. Again, observe how the water moves and reacts to the new temperature variable. What is the motion of the dye now? What oceanic processes that you've learned about do you observe? How does this differ from what you saw in the last activity? What other abiotic factors might affect ocean circulation? Now we can start to think about how these processes relate to the distribution of organic matter in the ocean. Here is a satellite picture of an algal bloom in the Atlantic Ocean. What do you notice about the dynamic patterns of this bloom? Based on what you know about the ocean productivity, where would you expect similar phytoplankton bloom events to occur? Thanks for watching, and I hope you enjoy the lab.